Good morning, students. So, in the process of genetic engineering, we have discussed the various enzymes needed to manipulate the DNA. Then next, we had discussed about the various vectors, that is the vehicle DNA, which can carry the DNA of interest into the host cell, and the third, of course, the host, in which we have discussed bacteria, that how bacteria can be made competent to take up DNA from outside and get transformed. And then the next host that we were discussing was plant cells. And plant cells, as we mentioned, that the most common way in which these are transformed, that is using Agrobacterium tumefaciens. Now this Agrobacterium tumefaciens, this one has TI plasmid, as I mentioned with you, TI plasmid, which is tumor inducing plasmid. Now in this TI plasmid, there is a region which is called tDNA, and it is this tDNA which gets integrated with the plant's chromosome, and it could be any chromosome, any arbitrary point at random, it will get integrated. And this tDNA codes for number of growth factors like auxins and cytokinins, and that's why there is crown gall disease occurs. So these are basically the genes in the tDNA we can say they are called phyto-oncogenes. You see, plant oncogenes we have discussed, the cancer genes. So here these are like as if in the plant the cancer is being caused, so has phyto-oncogenes which code for production of auxins and cytokinins. Auxins and cytokinins, and this results in crown gall disease. Crown gall disease. So the site, suppose this is the plant here, and in this plant wherever this entered, so because of that there will be abnormal growth taking place, and the part of this will be gall, basically is like a sore, so that will be projecting out. That's what is called crown gall. Now we need to use this ability of Agrobacterium to integrate within the plant's genome, but at the same time we do not want the plant to be infected. So what is being done? Of course, we are manipulating here at the tDNA. So we are removing some of the genes, and there we are putting the gene of interest, and this modified Agrobacterium is then allowed to infect the plant. And as I told you, this Agrobacterium it naturally infects only dicot plants, but it has been manipulated, that we'll see in golden rice in the next chapter, where it was manipulated so that modified so that it could infect monocot as well, but naturally it infects dicots. Okay, so naturally infects dicots. Okay, so in this case, as we said, that once again this will be called disarmed. So Agrobacterium is modified with the disarmed TI plasmid. Agrobacterium is modified with disarmed TI plasmid and allowed to infect the plant host. Okay, so now what will happen? The gene of interest also gets incorporated. So this is the most common method, you see. Apart from this, we can use gene gun. So next is gene gun, or which is also called biolistics. So here simply what is being done is the gene of interest is directly shot into the plant cells. So in this case, this gene gun, this is an apparatus in which minute micro pellets or they are taken, which are made up of tungsten or gold. Okay, so micro projectiles microprojectiles of tungsten or gold are coated with DNA of interest and shot into the plant cells. Now then these plant cells will be seen which one has taken the DNA. So basically now this is vectorless transfer. Here with this DNA of interest is not incorporated with the vector DNA. So this is vectorless transfer, okay, vectorless transfer, and in whichever the DNA of interest has incorporated, that cells we can separate out. So this is done in a culture medium, you see. So we can, just in short I can show you here, like suppose this is just a slide. On this slide we have kept the plant cells here, okay, and over this here we have the gene gun. Let's just put attach on a stand, okay, and here these micro projectiles are there, and there is a cartridge chamber and then a firing pin. So once it is fired, these tiny projectiles, they'll pass through the plant cell from one side to the other, they'll just go out. But on those projectiles, the DNA of interest which is present, that will remain, some of it will remain inside the cell and some of it can get integrated into the genome. And then of course we'll have to use probes, and we have to check out that whether which cells have taken up the DNA of interest, and those then we can put in a culture medium and make copies of it. Okay, so that is one method. And then of course using plant viruses, another method is there, using plant viruses. This is although not so common, and in these the viruses which have been studied for this purpose, those are Gemini virus and Columovirus. This one is otherwise also called cauliflower mosaic virus. Cauliflower mosaic virus. This is there because viruses also have the ability to integrate their DNA with the host DNA. So these are also being worked upon, but most common method, as I said, is agrobacterium, and the next one of course is gene gun. Now moving on to next host, that is the animal cells. Animal cells as host. So next one we can mention animal cells. In animal cells, of course, the most common method is using disarmed viruses. Okay, so one is using disarmed viruses, and the viruses of choice, those are retrovirus, because retrovirus in HIV you have seen that they have the ability to go into the host cell, and from their DNA, from their RNA they will form DNA, and that DNA will then integrate with the host DNA. So retroviruses have that ability, and yes, Ashish, you had asked this question yesterday. So retroviruses are animal viruses, so that won't be infecting the plant cells. Okay, retrovirus is there. Then there is adenovirus. This also has this ability. Adenovirus. There is papillomavirus. Papillomavirus. Even the herpes virus. So all these, of course, their virulent genes have to be removed and replaced by the gene of interest. Okay. Herpes virus. These all have been associated. Now in this adenovirus there has been a question also. This is a DNA virus, you see. So it's only the retrovirus which is a RNA virus. The remaining three that I have mentioned, they are DNA viruses, and they have the ability to integrate their DNA with the host DNA. Okay, so this is one. Then another method is Another method is the using microinjection. Now this is once again a vectorless method. Vectorless method of gene transfer. So in this case, but of course in this only one cell at a time can be transformed. So suppose this is the slide once again, we have this cell here, and in this cell directly into this nucleus by using a microinjection or a micropipette, we are injecting the DNA of interest directly. So this is the microinjection, and there is DNA of interest injected. So only one cell at a time can be done. Then although it's not there in the NCERT, but you can know it using, that is also vectorless method, using liposomes. Now liposomes are simply vesicles. Vesicles, you know, that's a small bag-like structure, and in this bag the DNA of interest can be loaded. The DNA is here, okay, and this vesicle is lined by phospholipid bilayer. So that's why you see liposome lipid body. Okay, this is how it would be, and this liposome can be made to fuse. Now since we don't have cell wall, so it can directly fuse with the cell membrane, and as a result the DNA will be going in, and some of the DNA can integrate once again with the host DNA. So this is so in this case lipid vesicles loaded. There is, just a sec, I have an important call. Yes, so lipid vesicles loaded with DNA, that is there, and this is then allowed to fuse with the host cell. So this is also vectorless method of gene transfer. Okay, and when we transfer gene or DNA of interest into the animal cell, especially using viruses, you may come across a term that is called transfection. Now transfection, because that is infection into the cell and that is transforming it. So we have combined two terms, that is transformation plus infection, that is called transfection. Okay, so anyway, now we have seen the three types of host, that is the bacterial cell, the plant cell and the animal cell, and what are the various methods of choice for the gene transfer, right? Now coming to what are the various steps, let us first list out some of the steps in proper order which are required for making a recombinant DNA. Okay, so steps for recombinant DNA technology, and this can also be called RDT. So the first step is, of course, isolation of DNA. Now this is basically when we say DNA, so this is whole DNA. We have to break open the cell, we'll come to that. Then restriction, digestion, separation of fragments, then amplification of DNA of interest, then of course ligation with vector, transformation of host, and then screening. Okay, so let us start with isolation of whole DNA. So first step. Now in this step, first thing that is to be done is that we have to break open the cell because the DNA is inside the cell. So first one, that is, as I said, isolation of whole DNA. Isolation of whole DNA. So in this, the target cells need to be broken open. So in this, there is enzymatic cleavage can be done. Enzymatic cleavage of cell wall. For this, if we have bacterial cells from which we want to take out the DNA. So from the bacterial cell, you know the cell wall is peptidoglycan and which is digested by lysozyme. So we will be using lysozyme here. If it is fungal cells, so fungal cells have a cell wall of chitin. So here we would be using chitinase. And if it is plant cells, then we would be using cellulase. Okay. So here we had to break open the cell wall, mind it. The cell membrane, for that it's not difficult because we can just keep the cells in water, and what will happen? Water will go in by osmosis and the cell will rupture. So for the animal cells, we can simply give them osmotic shock. By that we can break them. Okay, or otherwise usually for here also, because beneath the cell wall there is cell membrane, so usually they are given they are put in a solution in which some detergent is added. Now the detergent also, it denatures the membrane proteins and that membrane becomes weak and easily gets ruptured. So you see it is mentioned for cell wall, but along with that one more thing we can add on, that in the solution where this breakdown we are carrying out, in that solution we have the enzyme plus detergent. So detergent will cause the breakdown of the cell membrane, right? The cell membrane will break like that, and all this DNA will come out into the solution. Now this, it is not only the DNA which will come out, because when the cell breaks open, the other macromolecules are also there, proteins are there, RNA is also there. So they have to be removed. So in this case, the proteins are digested by adding proteases. So proteins digested by proteases, and RNA digested by RNase. Now DNA is left intact. Now that DNA is then precipitated by putting, DNA is precipitated by pouring chilled ethanol. Chilled ethanol. So in this case, the solution that we are having, in this solution, these all digested end products are there, DNA is intact. So in this, when chilled ethanol is poured, this chilled ethanol will be here, although ethanol and water mixes, you know that, but since it is cold and we put it very slowly from the sides, there would be immediate mixing, won't be there. And where the mixing is occurring at this interface, that is the watery solution and the ethanol, where they are mixing at this interface, lot of these white threads which are easily visible by naked eyes, these will be precipitating out. So it is at this interface that DNA will precipitate out. And as it is given in the NCERT, that they put a glass rod here and just roll it, and the DNA is taken out. That's called spooling. Okay, so DNA is extracted by spooling from this. Okay, so just a glass rod which is rotated and it will wrap on it. So now we have got the whole DNA. Now this whole DNA, you see, it's very long. You must have studied that even in the human genome, that if we put our DNA end to end, how long it is, right? So it is very difficult to operate it, means work on it. So that's why it has to be first broken into smaller fragments by using restriction enzyme, right? So that is the next step, so that we can easily manipulate it. So the second step is restriction digestion. So here the whole DNA is cut into smaller fragments, by restriction enzyme. Okay, now suppose, I'll put it here only. Suppose this is the entire DNA, and in this entire DNA we use, let us say, EcoR1. Now the site of EcoR1, the restriction site, can be randomly present. So suppose one site is here, one site is here, and the EcoR1 will act on this and we will get 3 fragments like this. So 3 fragments would be obtained, and since these fragments are obtained by restriction digestion, these are called restriction fragments. Now these restriction fragments, I hope you might have studied RFLIP, restriction fragment length polymorphism, because if suppose we are using DNA of 2 people and their DNA we have extracted and both of them we cut with the same restriction enzyme like here. So the total DNA, even though if it is same, but it is quite possible that there has been a mutation, because you see the site for EcoR1 is GAATTC. Now even if one nucleotide changes and these point mutations are continuously taking place, so then EcoR1 won't be able to recognize it. So let us say that these 3 fragments that we have obtained, A, B, C, in this person, the next person, this one has undergone a mutation. So what happens, that when we use this EcoR1, it will be cut only into 2 fragments. So there will be A, this one will be a longer fragment, B plus C. Okay, so that's what is polymorphism, because the length of the restriction fragments varies from person to person due to these mutations which are taking place, right? So that's what then that's what is RFLP, and these fragments we can separate. That would be our next step, that is separation of the fragments, that is by agarose gel electrophoresis. So now let us take this step. So separation of restriction fragments by agarose gel electrophoresis, that is H. Okay. So in this, DNA is negatively charged, right? We know that because of the phosphates, DNA is negatively charged, and any charged biomolecule if we have, we can separate it by electrophoresis, in which electrophoresis, by simple, if we go by the definition, it is separation of electrically charged molecules when they are placed in a externally applied electric field. Okay, so in this case, but this molecule requires a medium to move, and the medium here is agarose, Agarose, which is the agar that we know extracted from red algae. So that agar, just like in starch I had discussed with you, that starch occurs in two forms, that is amylose and amylopectin. Same way agar also exists in two forms, that is agarose and agaropectin. So agarose, this is a straight chain compound, right? And this agarose, when we this is in the labs, it is available in the form of fine powder, and they have to first dissolve it in a buffer solution. It doesn't dissolve because it's a mucopolysaccharide, you see, it doesn't dissolve in water just like that. So it will be put in a buffer solution, and then it has to be slightly heated also, then it dissolves, and then it is put in a tray, and when it cools, it sets. Just like jelly, it sets like that, and in that, when it is set, like suppose this is the agarose block, which is in a rectangular tray, it is put to set, and while it is setting, in this we put a substance here, that is a comb like thing, so that here the agarose doesn't come, and those will be small gaps which are called the wells. So the DNA that we have to move around, we will be loading it in this well. Now this entire agarose block will be put in a buffer solution. So you see, this is agarose block, which is a thick gel like substance, and in this, these are the wells in which DNA is loaded. So the electrophoresis unit, it's very simple. I'll just draw a very simple one. So here you see it is like this, and now I am giving a side figure, so that here it is the gel block, and this is the buffer solution, and from here we are putting to the electrodes, attaching it. Now since DNA is negatively charged, it is going to move towards positive electrode, so we have to place positive electrode here, negative electrode here. Now the DNA has been loaded, then the current will be switched on. This is buffer solution, and DNA will move through this agarose. So if we see this agarose microscopically, we will find it is fibrous mucopolysaccharide, and all these fibers, they are entwined so that there are some spaces in between, and through these spaces the DNA fragments are moving. And so we can very well understand here that the larger the fragment is, that will have more resistance, it will not be able to move quickly, while the smaller fragments can easily move through these spaces, right? So these will be moving, and then so this the normal language we use is that you run the gel. That means you put the load the gel in the wells, and then you switch on the current, allow it to run, and then after certain time it is then switched off. Now we have to visualize the DNA. So in this case, for that, DNA as such is not visible. So for seeing that DNA, we take out this entire block and dip it in ethidium bromide. Now ethidium bromide is a intercalating dye. Okay, so we'll just mention all this. So to visualize DNA after running electrophoresis, so intercalating dye ethidium bromide is used. Now this ethidium bromide, you see, is intercalating. Why did I say? That is between the DNA strands. Like you know, DNA is double stranded, so it will attach between the two strands. So wherever DNA is present, only that part will, the ethidium bromide will attach, and rest of the gel would be without this any kind of ethidium bromide there. Still it is not visible. Now we have to put the block in a UV chamber. So from the bottom UV rays are passed, and above there is a glass through which the observer can see, but once again that is having UV filter so that we are not exposed to those UV rays. Those are highly carcinogenic, right? So wherever this intercalating dye is there, this is also a fluorochrome dye. Now fluorochrome dye are those which will absorb UV rays, but they will emit it in the visible spectrum. Okay, so it's intercalating dye, that was because of the nature of it. Then the other thing that we can mention, it is a fluorochrome. It's a fluorochrome which absorbs UV rays and emits light in visible range. In visible range. So what happens? Wherever the DNA is there, we will see orange band. So let us make this block once again. So in this block, you see, let us say these were the wells where we had loaded it. So what is done usually, that is for reference point of view, in one well it is undigested DNA, the whole DNA that is loaded, undigested DNA. And in the other wells, of course, DNA from source 1 DNA, just like I was telling you about, for that for one person we are getting 3 strands, for the other 2 strands, like that. This is from source 2 DNA. Okay, and this side, as we had seen, this was towards the negative side, this is towards the positive side, like this. The DNA has moved. Now since undigested DNA won't be moving much, so thick orange band will come here. Here we see certain bands, okay, and in the other one there was B plus C, it had come, you see, imagine we can put it like this, and let us say this is A here. Okay, so with the previous diagram, let us say, no, B was bigger, no, okay, B, A, C, and this one is B plus C, this is A, so on, like that. So you see, we'll be getting this band. So the smaller the band, the smaller the DNA, that will move the farthest. So smaller DNA fragment moves farthest, and the larger fragments remain behind. And by doing this again and again, people have come to know that if it is 1000 kb DNA, it will be at this position, 800 kb will be here, 600 here. So likewise, you will see in higher books, they mention this range also here on the sides, just like a table. So we get an idea that what amount, means what size of the DNA has moved up to what point. Now we have separated the fragments, and whichever fragment we need, we can cut it out from the gel, and that's what is called elution. So elution is that DNA of interest is cut through the gel and removed. Cut through the gel and removed. Now NCERT gives just like that. But how would you know that this is the DNA of interest that you want? So there is a long process in finding it out, and that is through we will be using probes and southern blotting and all that will be needed. Then we will come to know that which DNA we want. So southern blotting as such, you see what is there, that the DNA has to be first blotted over a nitrocellulose paper. So now in this sample here itself, I will just show you here what is done for southern blotting. So first of all, what we do is that we put NaOH. Buffer solution is there, but the block is first dipped in NaOH, and when it is dipped in NaOH, the 2 strands of the DNA, they separate. And because on the nitrocellulose paper we can get only single strand of DNA, so double strand won't be possible. So first we have to denature so that the 2 strands are separate. Now after that, after treating it with NaOH, we keep it in a buffer solution, and above this we put a nitrocellulose paper. Now this nitrocellulose paper is put here just above the block, and From here there is above it there are number of filter papers we'll put, okay, number of filter papers, and a weight then can be kept on it, and a small wick just like the thread that is there in the candle or anything that we burn, because through that the buffer will rise up. The buffer would be rising up towards the filter paper, and when this buffer is rising, it will be rising through the gel block also. It rises through the gel block as well, and the DNA which is present in between, that DNA also rises along with that, and this DNA, when it reaches to the surface, it attaches on the nitrocellulose paper. Okay, so I'll just label it. Here is the gel block. I think everybody knows that. This is gel block. This green one that I have shown, this is nitrocellulose paper. These are filter papers, and this is a wick here, and this is a weight, right? So this is how the arrangement is for Southern blotting. Southern blotting, a technique developed by EM Southern, and because of that, and with this technique, what is being done, from the gel we are transferring single stranded DNA onto the nitrocellulose paper. Now once it has come on the nitrocellulose paper, now we can look for the DNA that we want. So for that, like suppose we want, let us take the example of insulin only. Now in that case, although for eukaryotic it won't work, because eukaryotic DNA, you know, it is with introns in between. So let us take some other example, some bacterial DNA, let us say. Now that bacterial protein we have coded, that bacterial protein has been coded, and as a result we know the sequence, so we will make a complementary sequence. So complementary sequence is taken, and that complementary sequence is in the form of a single stranded RNA or a DNA that is once again attached to a fluorochrome or a radioisotope, and that will be put on it. So wherever it will find a complementary, there it will attach and a hybridization will occur, and from that we will come to know which is the DNA that is actually targeted. So for probes, actually I wanted to take up anyways, because molecular probes has been an important question, and means it has been repeatedly there in our exams. Okay, so molecular probes, what are these? So molecular probes, or simply probes, we can use the term. So these are single stranded RNA or single stranded DNA complementary to the target DNA. And then of course the probes are labeled with radioisotope or what is that, fluorochrome. So wherever it will attach, we'll come to know either radioactivity is being emitted, or when we see it in the UV light, there would be visible light, something will be available. So through that we'll find that which DNA is our target DNA. Then it is taken out. So NCERT has taken the easy way out. They have just said this, but this is what is done. So now you can imagine with such difficulty the target DNA we have found. Now every time taking out target DNA, that would be difficult. Rather, instead, once we have got our target DNA, we can amplify it, making several copies of it. And when PCR was not developed, till that time the only method of cloning that target DNA was that you ligate it with a vector, put it in the bacteria, and as the bacteria divides and as this vector makes its copy, we'll get the cloning of that gene. But now we have a method of vectorless method of making copies of the gene of interest, and that is through PCR. So next thing we can talk about, that is amplification of DNA of interest. Amplification of DNA of interest by PCR. polymerase chain reaction. A technique developed by Kary Mullis. And this year you can expect a question on Kary Mullis, because just in 2019 he died, and he received the 1993 Nobel Prize for his work. Okay, and in this earlier, this reaction was of course done one after the other. The researcher has to sit over there and do. Now it is fully automated. There are machines, PCR machines, which are called thermal cyclers. Thermal cyclers. So these are PCR, automated PCR machines. Automated PCR machines. And that's why it has revolutionized. Now even for crime and forensics it is used. So even very little amount of DNA is available, then too we can make copies of it, several copies in less time, and then it can be matched with the suspects. Right. So first thing, let us talk about what all is the requirements to do a PCR. So requirements. One is of course the DNA of interest, so the target DNA. Target DNA, which as we said, it can be very small quantity. Then another is we need primers. Now in DNA replication you have seen, there it was RNA primers. RNA primers, they started the whole process, but here these are artificially made primers, and these are DNA primers only. Okay, so DNA primers, which are about 10 to 18 nucleotides long, DNA with which are complementary to the target, So whatever target DNA we want to make copies of, the ends we'll have to first sequence. So that is one prerequisite, you can say. So the end of the DNA has to be sequenced, and only then the oligonucleotides, these primers, can be prepared. So this is one. Then next thing that is required is the DNA polymerase. So Taq polymerase is there, which is a heat stable, so it doesn't denature in high temperature, heat stable DNA polymerase. And this has been found from a hot spring bacteria, you know that, Thermus aquaticus, and this was found around 2000. So when Kary Mullis actually started with PCR, at that time it was a very expensive technique, because with every cycle the DNA polymerase was getting denatured, and then again new DNA polymerase was to be added. But with this heat stable DNA polymerase discovered, now the same enzyme can be used again and again, so the process has become easier and cost effective. Okay, so then this is one. Then next is one more thing, magnesium ions. Magnesium ions are the cofactor for this enzyme. Okay, then DNTPs. Now DNTPs are what? Nothing but nucleotides, because when DNA is being synthesized, the nucleotides would be needed, the deoxynucleotides, and these deoxynucleotides, when it is happening within the cell, then of course they are there in the cytoplasm. So whatever you see, when the food we take, the DNA, RNA, that also in the food gets digested, it is absorbed, and it is present in the cells. But here, since everything is being done outside the cell in vitro conditions, so here it has to be also provided from outside. And DNTPs would be DATP, and if you remember D, when we have put, that means the sugar here is the deoxyribose sugar. So DATPs, DGTPs, D CTPs and D TTPs. Okay. Now as you see in the DNA, when it is placed, there it is, we require DAMP, because in the DNA or RNA the nucleotide is in the form of monophosphate, it is not triphosphate. But here it is taken triphosphate because when it is attached to the DNA, at that time the 2-phosphate will be utilized for energy for the bond formation. So those 2-phosphate will remove, and in the DNA actually it will remain in the form of AMP, DAMP, right? So that is there. Then so this is all the requirements that is needed, and of course all is done in a buffer solution, so buffer solution we can mention. Now this PCR, it runs in cycles and each cycle has 3 steps. Okay, so PCR runs in cycles and each cycle has 3 steps. So first step is denaturation, or which can also be called melting of the DNA. Melting of the DNA. So in this case the target DNA is heated. Okay, so target DNA is heated to 94 degrees Celsius. Some books they do write 95, doesn't matter, 1 degree will not have much effect and you won't have 2 options so close, never. So but basically all the biotechnology books they mention 94. So 94 degrees Celsius, and as a result what happens, why we are taking up to that temperature, because beyond 60 the denaturation starts, but wherever there are GC sequences there are 3 hydrogen bonds, so that requires a higher temperature. So by putting this much of temperature the 2 strands will fully separate, okay, not that some portion has separated, some portion not. So in this case what happens, the hydrogen bonds break and the 2 strands separate. Hydrogen bonds break and 2 strands separate. Okay, so we can put here in the diagram. So you see, suppose this is our target DNA, and in this part we want to amplify. Okay, so this is the target DNA, and by heating to 94 degrees Celsius, what has happened, the 2 strands have separated. Okay, like this. Now we come to the second step. Now in the second step what is being done is annealing with primers. Okay, so second step is annealing with primers. So now what is done, the primers are added and the temperature is brought down, because only then hydrogen bonds will form again. Okay, so primers are added and temperature is lowered in between, in the range of 50 to 60 degree Celsius. Okay, so now you see what happens, that these primers they attach. Now primers are short sequences, so lesser number of hydrogen bonds have to be formed, so those will be the ones which will attach, not the whole DNA, this is much longer. So in this manner here, let us say here this is the primer 5 prime to 3 prime, and here this is the other primer 5 prime to 3 prime. So these you see are the primers, so they have annealed. Now These primers, as I told you, they are complementary to the target DNA, the ends of the target DNA, but they shouldn't be complementary to each other. That is important, because if they are complementary to each other, they will form double-stranded hybrids, and then the whole process will not be done. So they have to be complementary only to the target. Now comes the third step, that is extension of primers. Extension of primers. Now for this extension, we require what? Taq polymerase, DNTPs. So now here annealing of the primers, you see this was done at 50 to 60 degrees Celsius. Now this is still at denaturing levels, that is 72 degrees Celsius temperature is there, and there is now Taq polymerase added. Taq polymerase, its cofactor is there, and also DNTPs are added. All these are added. So now what happens? Now there is a DNA polymerase available. So these primers, they will start transferring the nucleotides one after the other, so that the chain elongates. And in this, what is going to happen is that this, because you know normal replication also you have studied, the chain elongates in 5' to 3' direction. So here also the same thing, it is elongating in 5' to 3' direction, and same way here also it is going in 5' to 3' direction. So how is it different from the actual DNA duplication? Because in that what had happened, there was a replication fork formed. So at one end the 5' to 3' was available, but on the other side it was not. That's why we had the lagging fragments or the Okazaki fragments over there. But here the entire DNA has been separated out, so both strands are getting replicated at the same speed. Okay, there is nothing like leading or lagging strand, both are leading now, right? And this is what is the speciality of PCR. Now, so at the end what we are getting 2 copies of our target DNA. So extension of the primers, I can just mention here that primers elongate or extend in 5' to 3' direction. Of course, as you see, under the effect of Taq polymerase, at high temperature it is working, and so at the end of the cycle, 2 DNA molecules are formed. 2 DNA. When I say molecules, so that means the entire DNA having 2 strands, that is formed. 2 DNA molecules are formed. Now once the 2 molecules are formed, again the cycle is repeated. So now we have, when this will go for the first step, that is denaturation, then now we will have 4 templates. Now there will be 4 templates. Here there were only 2 templates, now it is going to be 4 templates, and all those 4 templates will undergo the replication. So at the end of second cycle we will have 4 molecules of DNA. So the number of DNA molecules formed at the end of n number of cycles is 2 to the power n. So as many cycles, with the end of each cycle there is 2 molecules of DNA. So the number is doubling, you see, from 2 to 4, 4 to 8, 8 to 16, so on. So that is 2 to the power n, right? And that's why you will see in the NCERT that if this cycle is repeated for 30 times, 30 cycles are there. So 2 to the power 30, if you will calculate, that is around 1 billion. And one cycle is hardly taking 5 to 6 minutes. So you can see that in such a short period of time, we have 1 billion molecules of our target DNA, and at this much is not possible even if we had transferred this gene into bacteria. Even their rate of division is not that fast. Okay, so this PCR has really revolutionized the whole process of biotechnology, because very quickly, in just few hours, 2-3 hours, we can have so many copies of our target DNA. And since this technique is so sophisticated that even small amount of DNA, so that's why you see it finds lot of use in forensics, because even if there is a drop of blood, it came on the shirt of the person also, then to just from there that can be cut out, and in that drop there might be a WBC, and from that the DNA can be extracted out and its copies can be made, and then wherever we think that this person's DNA it might be, it can be compared with that, right? And even lot of fossils, in fossils the DNA mostly is lost, but even small amount of DNA if we do get, like mitochondrial DNA we do get. In one case study I was studying where from the teeth of the fossil the researcher was able to extract out from the pulp cavity some amount of DNA. Now once it is there, it can be made several copies of, and then it can be compared with the present populations, and that way we can see that how much changes have occurred over the period of time. So these kind of so PCR is not only in biotechnology, but it has found use in so many other fields as well. Okay, so this is there. Now you have got your DNA, copy of the DNA of interest is there. And one more thing let me relate, that's because it has appeared in a question, so let me relate it here. I think if you will see previous year's paper, there was one option, although it was not the answer, but there was option TA cloning, it was there. T/A they had mentioned TA cloning. So because you see this Taq polymerase, it is a polymerase undoubtedly, but it is also a terminal transferase. So at the end when it comes 5' to 3', it is coming to this end, and it will go a little ahead and it will put one adenine over there. So it will create one adenine overhang. So the PCR, the one which are formed, this can be called PCR products. So the PCR products already have a overhang over there, and so the vector to which we want to attach it, we require there a T overhang, and we needn't use a restriction enzyme if this is there possible, and we can directly attach it. So we can just mention a line here about terminal transferase activity of it. So Taq polymerase has a terminal transferase activity also, terminal transferase activity also. Okay, so it creates adenine overhang, it creates adenine overhang, and as a result PCR products can be directly ligated to vector with T overhang. Okay, and this is what you see. You will see, I think 2-3 years back only this question was there. So TA cloning, it was there in the option, although it was not the answer, but once it has appeared in the option, one should know what it is. Okay, so that is what is called TA cloning. Anyway, coming on to the next thing, that is next, most of the things I have already done with you, that once you have got your gene of interest amplified, we can now attach it with the vector, which suitable vector, whichever host we are going to use, accordingly that vector we will have to search for, and with that vector we will attach it by the use of ligases, and then we will have to transform the host, that we have to put that recombinant DNA into the host cell, and after that we have to screen that which host cell has been positively transformed. That means it has the recombinant DNA in it, right? So these all steps I have already done with you. Now once this is done, then what is the there are certain terms that we use, that is like the other day I was telling you about that the vector, I just use the term vector DNA, but these vectors can be cloning vectors or expression vectors. Okay, so let me just take this part, cloning vectors, and the other is expression vectors. That's why I left it at that time. Now, like you see, suppose we were using PBR 322, and in this PBR 322 we have ligated a gene of interest. Okay, so this is the foreign DNA which we have ligated in it. Now with this, when we put it into the bacterial host, so as the bacteria divides or as this plasmid replicates, the copies, more and more copies of the foreign DNA are also made. So that would be using a cloning vector. So cloning vector, this is used only to make copies of foreign DNA. Okay, that is there. But in the industry, like when we talk about humulin again, so there we require that this foreign DNA should express itself also. That means it should be making RNA, and that RNA will then translate to form the protein. So when expression is required, then we need something extra to do. So in that case, along with the foreign DNA, we will have to put another sequence here, and that sequence is a promoter. A promoter has to be there, because when there is a promoter, then this foreign DNA will express. Otherwise, without the promoter, it will not express. Then it becomes expression vectors. So you see, all expression vectors are cloning vectors, but all cloning vectors are not expression vectors. So we can next mention to obtain the gene product. Now we just don't want just the copy of the gene, we want to get the gene product also, that is the protein also. Okay, so to obtain gene product, a strong promoter is also ligated with the foreign DNA. Okay, so accordingly that becomes expression vector. That's the only difference, because this once again has been a term in the test. So biotechnology, you see, is one topic in which the questions, you cannot say that they are only and only from NCERT. There is slightly you have to go above, and that's what things I have been taking with you. Now this protein, when it is, or the gene product which is being formed, if it is formed, like you see, a human gene expressed in a bacteria, so that protein which is formed, that would be also called recombinant protein, and it has been produced in what? A host which is not the same species, so that is called heterologous host. Okay, so recombinant protein is thus formed, recombinant protein is thus formed in heterologous host. Okay, so recombinant protein, because this is a protein after making recombinant DNA, and it is being expressed in a different species than from where the gene of interest was taken. So that is, that host will be called heterologous host, right? So this is one. Then a little about upstream and downstream. Okay, so upstream processing let us take first, and then I'll talk about downstreaming. So upstream processing. Now you see, upstream processing is nothing but upscaling, that we are going from a small level to a industrial scale. That's what is upstream processing. So this is basically upscaling. Now in this, you see, we started with getting the bacterial colony on a petri dish. So here was your recombinant colony, okay, on a petri dish. So this is the recombinant colony. Now this recombinant colony, we are going to transfer it into a nutrient medium, a culture medium, so that we can see whether it is finding that culture medium suitable or not. We can study that which type of nutrients it would be requiring more to show maximum growth, so on. So it is transferred into a flask, and these are shake flask. Shake flask because the nutrients should not settle at one end, so it is kept on a, basically there's a electrical stage or kind of thing which is slightly agitating continuously, so that even mixing of the nutrients is taking place, and in that we have transferred. So this is the nutrient culture medium here, nutrient culture medium, and of course I put a cotton wool here. Now once we have checked that it is nutrient culture medium is okay with it, and then next what we have to do is we transfer it into a small bioreactor, a lab scale reactor, maybe having 100 liter capacity or so. Okay, so from here to lab scale bioreactor, this is simple, because you see these nutrient culture mediums are very expensive. So that's why this proper checking, quality control is needed. So now we transfer it to this. Now we have checked all the conditions, everything is okay, and then finally it will go into the bioreactor. Now bioreactors, which are industrial scale bioreactors, this is also bioreactor, but industrial scale bioreactors, those are of 1000 liters or maybe more than that capacity, huge steel tanks. You might have seen that in the there is a photograph given in your microbes chapter. You will see it one after the other. Those steel tanks, those are those are actually those industrial scale bioreactors. So now from here, industrial scale bioreactors, to this it is going to be transferred. So this going from a small petri dish to this level where the capacity, as I said, 1000 liter or above. So that's what is upstreaming. Okay, so this is upstreaming. Now a little about these reactors, bioreactors. Now these bioreactors, these are basically you can say large tanks. Okay, so these are stainless steel large tanks in which the microbes are cultured for industrial production, okay, and here the conditions are regulated. Okay, so there are controls for temperature, pH, and foam removal, because you see, because of their microbial growth, they are releasing certain proteinaceous substance also in the surrounding medium, which forms lot of foam on the surface, and once foam is formed, the aeration will stop, circulation of the air will stop, and that's why the microbes will start dying. So there has to be continuous foam removal also is needed, and usually for that pine oil and all are mixed, so that the upper surface of the medium will have that oily layer, and there this foam will not form. Okay, so foam removal is there, and then aseptic conditions are maintained. Okay, so that other microbes, undesired microbes, don't go into it. Aseptic conditions. And of course, before starting with the transferring the nutrient medium and all, it is fully sterilized nicely by steam. It's not just hot water, steam, because steam, you know, has much higher temperature, so by that it is sterilized. Okay, so before sterilization by steam is carried out before starting with the process. Before starting the process. Okay. So you will see in your NCERT 2 reactors they have mentioned, that is stirred tank reactor, and there was a question also that why is that stirrer needed. The purpose is same which I told you in the shake flask, that is there is even distribution of the nutrients. So just a very simple form we can make it here. So you see, let us say this is the reactor, and in this there is a steam inlet through which the steam will be passed before starting with everything, and then the knob is closed. Here this is the nutrient culture medium, there is a stirrer here, and to this stirrer these fan blades are attached, which is called the impeller. That would be to further help in the mixing. Okay, so there is a pressure, there is a thermometer attached, so from outside itself the researcher can read the temperature, pH. Even there is a pressure gauge, because these microbes when they are respiring they will be releasing carbon dioxide, so a lot of carbon dioxide will get accumulated here, and so that has to be released from outside, because otherwise the pressure will increase a lot. And then there are sample ports to which the medium can be put and taken out. Okay, so these are few things, these are simple common things which we can mention. So here this is, as I said, this is the stirrer, this is pressure gauge, this is temperature, this is pH, this is impeller, nutrient medium, sample port, this is steam inlet, right? These are there. So this is one, and the other one that they have given is the sparged tank reactor. Sparged tank reactor. Now this sparged tank reactor is just one difference basically. In this there is a sparger. So this is a sparger here. And through this sparger, this all will be there, everything. Air, of course this is sterilized air, that will be introduced. So this would be for those microbes if the microbe is aerobic. If it is anaerobic, we don't need it. If it is aerobic, so we'll have to pass sterilized air, oxygen, and this will be through the sparger. There are holes in it, and through this air will be added to the mixture, right? That's the difference basically. So this is one. Then this culture medium which we are taking, this culture medium or the culture media, we can do this also we can categorize into three types basically on the basis of culture media. One is called the batch culture, another one is called the continuous culture, and the third one is called fed batch culture. Okay, now batch culture is simply that we clean out the entire fermenter. This is also called biofermenter. We clean it out, and then we take a certain amount of the nutrient medium in it, inoculate it with the microbe, and then close it down, means further new amount of nutrient will not be added. So that's why it is called a closed culture also, and allow them to carry out their process. So once the product is formed, that we can test from the sample pot, and then the whole thing will have to be taken out, because you see, like for any growth curve, as you see that it will initially there is a lag phase because they get acclimatized to the new culture, and then there is exponential growth, and after that once the medium they are using up, they have used up, so there will be a stationary phase, and then finally a declining phase will also start, they'll start dying. Like you see in case of alcohol production, you must have studied that in microbes. In alcohol production, the yeast, after the level of alcohol reaches a certain level, the yeast start dying. So when they have started dying, there's no point in keeping them further. So we just have to take out the entire thing, separate out the alcohol, and the amount of alcohol is then increased by fractional distillation. So there batch culture is used, where the culture medium is of a constant amount. Okay, so constant amount of culture medium is taken, culture medium used, and it is removed when the stationary phase is reached, and it is removed on reaching stationary phase. Okay, so that is in case of batch culture. And as I said, alcoholic drinks, they are prepared by batch culture only. Now coming to continuous culture, in continuous culture we are maintaining the exponential phase. This exponential phase is maintained. So here this one has been a question once again in our NEET exam. So exponential phase is maintained. Exponential phase is maintained, and how it is maintained, that here we take out some amount of the culture medium, a particular volume, and which has been used up by the microbes, the nutrients have been used up, and that will be replaced by a fresh volume. So here we can just mention, so that's why it is called open culture. Okay, continuous culture, this is open culture, I'll write it here. So in this case, used culture medium is removed and replaced by fresh culture, medium. So as a result, exponential phase is maintained. But there is nothing like that it will go on and on for years. It can go on certainly more than the batch culture, but usually every time we open and put fresh volume, there is always a chance that some infection can creep in. And once that happens, then once again this also has to be stopped. The whole thing has to be taken out. The entire bioreactor has to be cleaned properly, and it has to be started afresh. Okay, but still it can go on for long duration. And then coming to the last one, this is an intermediate approach. Intermediate approach between the batch and the continuous culture, fed batch culture. In this, the fresh medium is added, but the used medium is not removed. So fresh medium can be added up to certain level only. Beyond that, there is no capacity, and after that we'll have to then remove and get the process closed for the time being. So here fresh culture is added. Fresh culture is added without removing used culture. Without removing used culture. Okay. So this is more commonly used, the fed batch culture. Now the last step, the product has been formed, let us say. So we have to isolate that product. Now comes the payoff phase actually. After that too. So suppose there were bacteria here, our microbe was a bacteria. Now bacteria could have made the protein which is present within the bacteria, or it may have secreted it here into the medium. So wherever it is, we have to extract it. And out of so many different chemicals which are present in the bacterial cytoplasm, also so many chemicals are there, we have to take out that particular product of interest. Now we are talking about product of interest. So that is what is downstream processing. Okay, downstream processing. Okay, Now this downstream processing, this is the, in this case, the product of interest is isolated, purified, and if some modification is required, it would be modified, then it is subjected to rigorous, like medicines and all are there, so there is lot of testing required, so subjected to rigorous quality control, and finally marketed. Now it is ready to go in the market and finally marketed. Like every day nowadays, you see every day there is a news, some or the other, that there has been a vaccine prepared to protect from corona. I wish everybody knows a little biology and people read biology, because you see even if the vaccine is prepared, which the chances are not there, so that will have to be, it cannot easily just be, it will not be coming in the market. It will have to be properly purified, because it should be such that some side effects are not there. We'll have to check that, and then it is tested on patients who are already terminally ill, like cancer patients and all are there, who give this kind of you can say consent that their body can be utilized for testing, and then it is then tested on actual patients, and then finally after this it will be available for others to use. So it's not so easy that one suddenly it will come out in the market. So anyway, this is one, and then this is the most labor intensive step you see. So in any biotechnological firm, the major workforce is in the downstream department. Okay, so this one is the most labor intensive step. Okay, and in this number of things are done. You see there is filtration, there is electrophoresis, chromatography, lot many techniques are used. Okay, so requires use of filtration, electrophoresis, chromatography, etc. Okay, so many, because even in nanograms, if some impurity is there, that would be having a dangerous effect. So this is a very, very important step which the company has to go through with. So this is how the entire process works, and I think with this we have completed our topic. There hasn't been any questions today, any doubts. If you want, you can ask here itself, because the chapter is yet fresh, so you can put up your questions, and so we can deal with them before we move on to next chapter. Yes, students, the class has been too quiet today. Yes, anything coming up? Okay, then let me ask one question. Do you know what is reporter enzyme. Yes. Anybody? So, like here you see the case we had seen in blue-white screening, that was beta galactosidase enzyme. Beta galactosidase enzyme. So, the enzyme by which we come to know about the positive transformants, that means where the transformation has been correct, that is recombinant DNA has gone in, that enzyme will be called reporter enzyme, right? As simple as that. So reporter enzyme, so the enzyme that gives information about the positive transformants, and positive transformants we can use the term recombinants, that is called reporter enzyme. Okay, right, here we have, it is used as a selectable marker. Yes, in a way you have got it. Ashutosh, I think you have gone a little beyond. Used for expression of transgene in target tissue. I think with that better would be that it is a kind of selective marker only by which we come to know whether the host cell has got transformed or not. Okay, that would be a better answer. So almost yes, you have got the thing. So do go through the questions, and as I told you, biotechnology, so see the previous year papers also. It's a very important topic, and so next topic we'll be starting with is biotechnology and its applications. It's almost time, 3 minutes left, so I can wind up the class. So more or less we have come to the last chapter of the syllabus finally. Okay, so thank you students.